And today I'm going to talk about an emerging role of FUS at the neuromuscular junction and its implications for ALS and FTD pathogenesis. So back in 2003, we identified a large multi-generational British family with ALS, and we linked this kindred to the region on chromosome 16q, and this region is also shared by other families, so which confirm this linkage. So we started some sequencing of some candidate genes, and in 2009, Caroline Vance in our group, she identified this R521C mutation in exon 15 of FUS in this kindred. And so this mutation caused an arginine to cysteine substitution at position 521. And this mutation was also identified in another family. And in addition, in the same position 521, another mutation, R521H, also identified in two other families. And a third mutation, R514G, was then identified. And so these are like the first mutations that Caroline published, which cause familial ALS. So FUS is a DNA and RNA binding protein involved in RNA processing, and the mutation occurs in about 4% of familial ALS, rarely sporadic and in frontotemporal dementia. So patients carrying these mutations, they develop inclusions in their motor neurons. And as you can see here that there are a lot of other mutations identified in this protein, FUS. However, the majority of them lie within this nuclear localizing signal at the C-terminal. And also study shows that mutant FUS can also sequester wild-type FUS and cause aggregation. So here you can see this FUS aggregates in ALS due to FUS mutations. So these are these spinal cord motor neurons in patients suffer from with these ALS mutations. And you can see these large globular and elongated FUS inclusions in these patients, which were absent in the control, in the sort 1 familial ALS, and also in the sporadic ALS. And at the bottom, you can see these motor neurons with large FUS inclusions appear to have nuclear clearing of FUS. In addition to familial ALS, FUS pathology is also identified in a subset of frontotemporal dementia with ubiquity inclusions, and also in neuronal intermediate filament inclusion disease. So this is like FUS staining in some postmortem brain tissues from patients. So normally this is the control. You can see FUS staining, which shows a typical nuclear localization of FUS. However, in these patients, FUS was identified as the component of the neuronal cytoplasmic inclusions, also the intranuclear inclusions at the bottom. And FUS inclusions were also identified in BIBD, which is the tau negative form of FTLD. So this reinforced the concept that ALS and FTD are closely related. So our studies also show that the C-terminal of FUS contains the nuclear localizing signals. So here you can see this, the transfected CV1 cells. So we show that the GFP-tagged wild-type FUS shows the nuclear localization, which localized with DAPI. And in the FUS mutant, this shows an increased cytoplasmic localization. And in this truncated FUS protein, in which the C-terminal, the 17 amino acids which contain the NLS were deleted. So in this truncated FUS, there is almost an exclusive cytoplasmic staining. So this confirms that the C-terminal of FUS does contain the NLS. And in order to show that, or in order to determine how come this mutant FUS has this cytoplasmic localization, so we then insert this 17 C-terminal amino acid, which contain the wild-type NLS, to the C-terminal of the mutant FUS. And as you can see here, that there is a restore of this FUS nuclear localization. So this shows that mutation or deletion without the NLS caused impairment in nuclear import of FUS, and that when we add the functional NLS back to the mutant FUS, it rescues these phenotypes. And we further shows that the mutant FUS mislocalize in fibroblasts to form these stress granules. So we also look at patient fibroblasts. So you can see that in the mutant fibroblasts, there is a significant increase of this cytoplasmic staining of FUS. And when we add arsenite treatment, which causes oxidative stress to these fibroblasts, there is a significant increase of this PABP-labeled stress granules in the controls and also in the mutant FUS. However, only in the mutant fibroblasts that we find this significant increase of FUS-positive granules, as you can see here. So this shows that this cytoplasmic FUS was recruited to the stress granules and colocalizing here. So this suggests that the aggregation of FUS in the stress granules could seed oligomeric aggregate formation, and this may impair the RNA processing and cause neurodegeneration. So despite the fact that the best described functions of FUS is the regulation of transcription and splicing in the nucleus, however, a lot of recent studies have shown that FUS is involved in mRNA transport, stability, and local translation, which include the binding and shuttering of mRNA into synapse. For example, this target ND1L mRNA, studies have shown this FUS transport this mRNA into the dendrites in different cells, and this is involved in the actin cytoskeleton reorganization in spines. And also the localization of FUS in the dendritic spines were shown as RNA granules. So this suggests that maybe there are some specific cytoplasmic function of FUS, and the impairment of this function may cause this regulation of axonal transport and also some deficits at the synapse. So in our lab, we also try to study FUS pathology using iPS cells. So Carol Shum in our group, she is leading this study. So she managed to characterize a lot of these FUS iPS cells and also differentiate them into neurostem cells and motor neurons. So here, she found that in these iPS cell-derived motor neurons, FUS is present in the axon, so as you can see here. And this axonal staining was dramatically increased in the mutant motor neurons. And she further characterized the dendritic aberration, and she found this dramatic loss of dendritic protrusions in these mutant motor neurons with shorter neurites and reduced branches and processes. So this suggests that FUS has a function in the dendritic spines, and maybe FUS mutation cause some dysfunction in the synapse. So in ALS and also some other neurodegenerative diseases, dysfunction in the synapse and also the peripheral neuromuscular junction have been observed in very early disease stages before the onset of symptoms and loss of motor neurons. So this suggests this dying back neuronal degeneration. And there are a number of suggested mechanisms in this causing this dying back phenomenon. For example, disrupted axonal transport and neurofilament assembly, and also muscle hypermetabolism leading to energy deficits, and also maybe some changes in the gene expression of neurotrophic factors and synaptic proteins. So as you can see here on the left, so normally transport and communication occurs normally via the axon connecting the spinal cord motor neurons and the peripheral neuromuscular junction in the muscle end plates. And in the dying back phenomenon, neuromuscular junction degeneration occurs first, which actively causes axonal degeneration and then back up to the motor neuron degeneration in the spinal cords. So there are quite a lot of studies in ALS patients and some animal models, which shows that the neuromuscular junction pathology was found way before loss of motor neurons. And more recently, there are a lot of FUS transgenic models which have implicated synaptic dysfunction at the NMJ. This including studies in Drosophila and also in zebrafish. So these studies together shows that the NMJ pathological changes occur before they found loss or some change in the motor neuron, and they also implicated FUS may have a specific function in regulating the synaptic transmission in these models. So these together imply that the peripheral function of FUS, which impairment of this function may cause dysregulation of transport and synaptic dysfunction, which cause this FUS-mediated ALS. So I was very interested to look at the role of FUS at the neuromuscular junction, and so the transgenic models I used are these transgenic mouse models of ALS, which overexpress the human wild-type FUS. So these mice were generated by Jackie Mitchell in our group, which she published these mice last year. So in the homozygous mice, these mice have aggressive phenotype with an early onset tremor by 4 weeks, followed by the progressive hind limb paralysis and death by 12 weeks. And you can see here, these homozygous mice are typically like much smaller compared to the non-transgenic littermates. And also they show this abnormal hind limb splay when lifted up by tail. However, in the hemizygous mice, no motor phenotype or pathology was found. And when we look at the Western blot of the brain lysate, it shows that there is a dose-dependent increase in the hard-tag human FUS expression in the transgenic mice, and also a mild decrease in the endogenous mouse FUS due to autoregulation. And so this shows that the overexpression of human FUS downregulates the endogenous FUS expression. And also, these nuclear and cytoplasmic fractions from brain shows that there is an increase of cytoplasmic FUS levels only in the homozygous mice, as you can see here. And so in these homozygous mice, there are a number of clinical phenotypes shown. So these mice develop motor dysfunction from 4 weeks onwards, and they are dead by around 10 to 15 weeks. So here you can see that in red, this is the homozygous mice. You can see that from 4 weeks onward, they fail to gain weight, which was the really small mice, as you can see before. And also they perform really badly on the rotor test. And so they start to have a decline of this motor function at 4 weeks, and also a dramatic loss of the locomotor activity in an open field environment. So these mice die around 82 days. Now I'm going to show you just a very short video. So here on the left, you can see this is the normal non-transgenic mouse at 8 weeks. So you can see it's like really happy, like walking around and exploring the environment. So now I'm going to show you the video on the right, which is the So this is the homozygous mouse at 8 weeks. So you can see that it has developed a very severe tremor and locomotor dysfunction. So these mice have increased hind limb paralysis and also a very severe stilted gait. So they are unable to raise their pelvis off the ground. And normally these mice require to be euthanized by about 12 weeks because they have lost more than 25% of their body weight. And they can't really access food or water. So this is a very severe phenotype. So in this end-stage homozygous mice, we also look at the loss of motor neurons in their spinal cords. So this is the non-transgenic spinal cord and the hemizygous. So you can see there is not much difference between them. However, in these homozygous end-stage mice, you can see that they have lost about 60% of their motor neurons and associated with muscle atrophy and also impaired neuromuscular function. And we also look at the spinal cord and muscle pathology. So we find that these homozygous mice show these FUS inclusions in the spinal cord, and increase in astrogliosis, and also muscle atrophy at 8 weeks old. So you can see here, this is the H&E staining. You can see this grouped and scattered muscle fiber atrophy only in these homozygous mice. So one very interesting thing is that although apart from all these pathological changes that we found in the spinal cord and muscle, there is actually no loss of neurons and very little neuroinflammation detected in their brain. So this may suggest that the neuronal degeneration in these mice may follow a dying back pattern. So I was very interested to try to characterize any changes at the neuromuscular junction in these mice. So I decided to perform an in vivo study of the axonal and synaptic degeneration at both presynaptic and symptomatic time points along the disease progression. So I looked at the NMJ morphology and also tried to localize FUS in this peripheral NMJ, and also looking at some pre- and postsynaptic proteins. So in the mice, the establishment of fully mature NMJ occurs during the first 3 weeks postnatal. So here you can see this adult neuromuscular junction. So this is the nerve terminal and the junctional folds at the muscle end plates. So acetylcholine receptors are enriched in these junctional folds, which can be stained by alpha-pancreatoxins, which is shown here in red. So this is the typical adult NMJ, which shows this typical pretzel shape because of the junctional fold. So this shows the development of the NMJ in the mice. So first, I look at 8-week-old young adult mice in both non-transgenic and transgenic mice. So I stain the gastrocnemius muscle with alpha-pancreatoxin, which stain the postsynaptic acetylcholine receptors, and with synaptophysin, which is the presynaptic marker of the NMJs, which stains the synaptic physical membranes at the nerve terminals. So you can see in the non-transgenic and hemizygous mice, which show no phenotype, there is a perfect colocalization of the pre- and postsynaptic markers. So this shows that these NMJs are fully innervated. However, only in the homozygous mice that I couldn't find the staining with synaptophysin. And also you can see the bronchotoxin staining here shows that they are much reduced in area and very narrow, which shows that the NMJs are denervated and also reduced in size in these mice. So I also quantified the denervation at the neuromuscular junction, and in these homozygous mice, about 90% of the NMJs are denervated at the end stage. So the next thing to look at is to try to localize FUS, and surprisingly, I found an abundance of FUS at the neuromuscular junction in these mice. So this is a non-transgenic normal mouse. And so I stained this with FUS and bronchotoxins. As you can see here, there is a perfect colocalization, which suggests that FUS is highly abundant at the neuromuscular junction. So this is very exciting indeed, because up to now, the localization of FUS at the NMJ has never been reported before. And I also tried to localize TDP43 and other FUS interacting proteins at the neuromuscular junction, but none of them colocalize with bronchotoxins like this. So this suggests that there may be a specific role of FUS at this peripheral neuromuscular synapse. So I stained this gastrocnemius muscle with FUS and bronchotoxin in both non-transgenic and transgenic mice. So at the end stage, again, in non-transgenic and hemizygous mice, there is perfect colocalization, which shows that FUS is highly abundant in these mice. However, in the homozygous mice, there is loss of FUS at the NMJ. And again, you can see the NMJ start to degenerate at this stage. So I was very interested to see and want to know, when does this loss of FUS occur, and also this NMJ degeneration, at what time point and when does this start? So I decided to go back and look at some very young postnatal mice. So first, I look at 1-week-old postnatal day 6 mice. And I found that FUS was abundance at the NMJs in both non-transgenic and hemizygous mice, including these homozygous mice. So you can see colocalization of FUS and bungarotoxin here. And so this shows you that I also tried staining with TDP43, and I couldn't find any staining which colocalized with bungarotoxin in all of the mice here. And also at postnatal day 6, all the MMJs are fully innervated, including the homozygous mice at day 6. So you can see this colocalization of pre- and post-synaptic markers. So then I go on to look at 2 weeks old postnatal day 15. And again, I found that FUS is still abundant in non-transgenic, hemizygous, and homozygous mice, as you can see here. And again, this shows us no TDP staining at all in the neuromuscular junction in these mice. And so when I look at the staining with synaptic physin, so in non-transgenic and hemizygous mice, so these NMJs are still fully innervated, as you can see here, which shows the staining colocalizing with bungarotoxin. However, in the homozygous mice at P15, I started to see some loss of synaptic physin at the NMJs, and also some reduced area of the NMJs in these mice. So this suggests that there is a loss of the pre-synaptic protein, synaptic physin, as early as postnatal day 15, which is very early pre-symptomatic stage. And one thing that's very interesting is also that at this stage, there is abundance of FUS in the homozygous mice, so which might suggest that overexpression of FUS may cause impairment or reduce the expression of these presynaptic proteins. So here it shows that the percentage of NMJ innervated by synaptic physin at P15. So you can see that there is an increase of partially and fully derivated NMJs in these mice compared to the non-transgenic. And I also tried to look at the NMJ area in these mice by just using MHA to measure the area stained by bungarotoxin. And at postnatal day 6, there is no difference in the size of the NMJ between non-transgenic and homozygous mice. However, at P15, I saw some reduced area of NMJ only in the homozygous mice. So this together suggests that at the presynaptic stage, P15, there was a loss of presynaptic protein synaptic physin and peripheral denervation, and also some changes in the NMJ morphology. So this again suggests that maybe in these homozygous mice, there is a dying back motor neuron degeneration. So at this point, it is very crucial to find out whether at this stage, is there any changes in the spinal cord. So I did some crystallographic staining of the spinal cord in these mice at postnatal day 15. And as you can see here, the non-transgenic and the homozygous mice here, there is actually no difference between them. So I didn't see any loss of spinal cord motor neuron at this stage. So this suggests that at the presynaptic stage, NMJ denervation and degeneration occur before any change or degeneration of motor neuron in the spinal cord, which suggests that there is a progressive dying back degeneration which occurs in the peripheral NMJ before motor neuron degeneration in the spinal cord. And this looks really promising, because here we're also suggesting that this dying back degeneration may be directly related to or caused by an impaired peripheral function of FUS, given that we found this abundance of FUS at the NMJ. Oops, sorry, press the wrong. So to come to my conclusions, so we found that FUS mislocalization and cytoplasmic aggregation are the hallmark pathologies in both ALS and FTD, which highlight the role of defective RNA processing in neurodegeneration. And recent studies and localization of FUS at the dendritic spines suggest that it has a role in axonal transport, peripheral translation and degradation of RNA. And in our mice, we find that overexpression of wild-type, human wild-type FUS in homozygous mice resulted in an aggressive and ultimately fatal motor deficit. And disruption at the NMJ was one of the earliest pathophysiological events that we found in these mice, which imply a dying back axonal degeneration mechanism. And also found that FUS is highly abundant at the NMJ in both non-transgenic and transgenic mice, which implicate that it might have a specific function at this peripheral synapse. And found that at P15, this pre-symptomatic stage, there is a loss of presynaptic protein, denervation, and also change in NMJ morphology in the homozygous mice. However, at this stage, there is no loss of spinal cord motor neurons. So this together suggests that there may be an emergent role of FUS at the NMJ, which regulates axonal transport or maybe also peripheral translation of the synaptic protein saprophysin at the NMJ. So our studies support that dying back disease mechanism, which impaired peripheral function of FUS, may actively cause synaptic and axonal degeneration, and then motor neuron degeneration up in the spinal cord. So further investigations, we needed to try to elucidate further this dying back neuropathy, and it will be interesting to identify any FUS interacting proteins which may be involved in this disease mechanism. So finally, I would like to thank all members in Shaw's lab for their hard work, and especially Caroline, Jackie, and Carol for their work in FUS, and especially Jackie for her help with giving advice and help with the mouse work, and of course all the funding bodies in supporting our work. So thank you very much.